In recent years, an emerging technology has begun to reshape the landscape of modern medicine. In 2022, the U.S. Food and Drug Administration approved a landmark policy allowing the use of microfluidic chips to replace certain forms of animal testing. This move represents not only a technological breakthrough, but also a major turning point in medical research. Across the medical field, clinicians have long hoped for faster, safer ways to bring new drugs to patients. However, under current regulations, the journey from laboratory discovery to an approved treatment is notoriously long and expensive. Spanning basic research, animal tests, large-scale preclinical trials, and 3 phases of clinical evaluation, the entire process can take up to 20 years. An enormous time and financial burden for researchers and pharmaceutical developers alike. The advent of organ-on-a-chip technology promises to change that. These micro-engineered devices simulate the behavior of human organs at the cellular and tissue levels. When a new drug is developed, scientists can use the chip's integrated sensors to quickly determine which diseases or conditions the drug may effectively target. This approach accelerates the identification of promising treatments while reducing dependence on animal testing. One of the technology's most significant advantages lies in its precision. By cultivating cancer cells or damaged tissues derived from actual patients on these chips, researchers can more accurately test the effects of nanoscale medicines. Because the cells originate from the human body, the experimental results are often more representative of real clinical outcomes. Integrating organ-on-a-chip technology with advances in nanomedicine not only reduces animal usage, but also allows for treatments that more closely reflect patient-specific conditions, ultimately improving therapeutic outcomes. A research team in Taiwan has applied this technology to the development of treatments for rare diseases such as osteosarcoma. Supported by the National Science and Technology Council, the team is focused on using flexible microfluidic chips to accelerate the discovery and application of rare disease drugs. Unlike conventional systems built on rigid materials such as glass or PMA, these new chips are made entirely from elastic materials, mimicking the softness of living human tissues. This design enables realistic biological activity and supports 3-dimensional cell culture, offering more reliable data than traditional 2-dimensional methods. The development of these fully flexible chips relies on the integration of flexible electronics and semiconductor technologies. Researchers note that this novel manufacturing process requires further discussion, refinement, and interdisciplinary collaboration to overcome engineering challenges. The development of these fully flexible chips relies on the integration of flexible electronics and semiconductor technologies. Researchers note that this novel manufacturing process requires further discussion, refinement, and interdisciplinary collaboration to overcome engineering challenges. Organ-on-a-chip technology represents far more than a replacement for animal testing. It signals the beginning of a new medical paradigm. It brings greater speed, safety, and personalization to drug development and patient care. The future of medicine, it seems, is quietly taking shape on these tiny chips.